Good afternoon, everyone. I know that it's very close to lunchtime, so I will move on with my talk. So I'm from Xavier University of Louisiana, which is in New Orleans, and we are a HBCU, primarily undergraduate institution. And my focus of research has been on the design and development of new series of kinase inhibitors. So we all know that the human genome contains, it was 514 last year, but it's now 518 kinase genes. And protein phosphorylation is one of the main functions of these proteins. And it is generally frequently post-translational modifications that are happening. And these are regulatory proteins, and abnormal phosphorylation is the hallmark of many types of cancers, as well as other types of diseases, including neurodegenerative disorders, where you see the tauopathy being due to hyperphosphorylation of the tau protein, and other kinds of cardiovascular diseases and immunodeficiencies. There are activating mutations. There are also other inactivating mutations in these proteins. And several of the protein kinases have been targeted for cancer treatment. The one issue with the present therapeutics for the cancer treatment is that many of the patients develop resistance to the existing therapeutics, and then it becomes very difficult to treat patients with those kinds of resistance. So there is always a need for new kinase inhibitors that can function as anti-cancer agents. The other issue is that while select few kinases have been targeted for as therapeutics, there are actually more than 120 oncogenic kinases. Hardly around 15 to 20% of those kinases have been successfully targeted. There is potential for many more protein kinases to be targeted as cancer therapeutics. So generally, the basic three-dimensional structure of a kinase is like this, with the N-terminal domain, C-terminal domain, and this is the hinge region. That is where the ATP binds, and this end is the catalytic phosphotransfer region of the kinase. So one of the lead structures that have been used by many researchers is taurosporin. And this is a natural product, but it is a pan-kinase inhibitor. But structural features of taurosporin have been used by many research groups, and including me, where as a postdoctoral fellow at Georgetown University, I was able to identify two different molecules as specific kinase inhibitors. This is for the GSK3 beta kinase inhibitor. At present, it is in phase 2 clinical trials for pancreatic cancer. And this was a VEGF kinase inhibitor. It's being explored in preclinical studies for angiogenesis. So these were some of the successes that I had as a postdoctoral fellow. But coming back to being a faculty, I had to develop my own set of molecules, and I have been working on that over the past few years. So the basic premise for a kinase inhibitor is that it is an ATP competitive inhibitor. And the one critical feature of all these molecules is that they have to bind to the hinge region, and they have substituents on different parts of the molecule that can reach to the different parts of the binding site in the binding cavity of the protein. So while we have a core molecule that can bind to the hinge region, these substituents on different parts of the molecules are the ones that are going to dictate the specificity as well as potency of the kinase inhibitor. So we, in my lab, we have developed a couple of core structures that can be used to attach different substituents on different parts of the molecule, so that we can make it more specific for certain kinases, and at the same time target other residues in the binding cavity to increase the potency. So we have created 54 derivatives of the lead compounds. We are in the process of making many hundreds more of those derivatives. And some of the kinase targets at present is CK1 epsilon. We are getting highly selective isozyme specific inhibition for CK1 epsilon. So we are looking at inhibiting tau hyperphosphorylation for Alzheimer's and also the period proteins for sleep disorders. And P70S6K1 is a downstream target for mTAR pathway. So that we are looking at that for anti-cancer properties as well as diabetic therapeutics, and GSK3 beta for pancreatic cancer as well as Alzheimer's disease, because GSK3 beta also phosphorylates the tau protein. So we have a couple of agents that are inhibiting both CK1 epsilon as well as GSK3 beta. So the dual inhibition is a very good property. That way we can use less of a drug to achieve dual targeting. And these are the other different kinases that we have been able to target somewhat selectively. And we are at present working on increasing the potency of those molecules. So this is an example of where the P70S6K1 protein lies in the downstream of the mTAR-C1 pathway. As you can see, it has several functions in several different molecular pathways. And what I can show is that some of the compounds that we found were very specific for P70S6K1. At present, this is 0.4 IC50 value. I have other compounds that have shown higher potency for P70S6K1. And we were able to prove that it is indeed targeting that particular protein by looking at the downstream RPS6 phosphorylation using Western blot studies. And we were able to show that we are able to reduce the phosphorylation on RPS6 protein, which is responsible for amino acid synthesis during cell growth. The other for casein kinase 1 epsilon selectivity, we can clearly see that we were able to achieve almost 5-fold selectivity for casein kinase 1 epsilon over casein kinase 1 delta. And we have other compounds that have shown almost 15-fold selectivity for casein kinase 1 epsilon. So we are looking at the both the tau phosphorylation status with those compounds, and they have shown promise in inhibiting the tau phosphorylation, and also looking at the exon 10 expression, because exon 10 plays a role in managing the ratio between the 4R and 3R tau proteins in the brain. And any disruption in this ratio seems to be causing adverse effects in the neuronal cells. So we are looking at those issues in the biological effect. And of course, we have other kinds of molecules that have shown very good targeting, especially this particular molecule. It shows that it is not inhibiting the native KIT protein, but all the mutants are being targeted successfully by this particular molecule. And there is one that is also targeting multiple JAK isozymes. So cross isozyme inhibition is also one thing that needs to be seen, because other isozymes can take over the role of the isozyme that we are targeting. So targeting cross isozyme protein kinase inhibition is one of the favorable properties. And we are also looking at red kinase as one of the targets. We have also achieved nanomolar potency for PIM1 kinase and GSK3 beta kinase. There are several different type compounds have shown activity specific with a high potency for different types of kinases. So what I am looking for at present is that I do not have collaborations with that many people to test it out on different cancers. So I'm looking for collaborations and also a way to move this project forward in terms of toxicology evaluations and other pre-clinical studies that is required for IND. So I would like to thank Louisiana Cancer Research Center for providing funding for this project, and the BUILD, which has also funded me, and my postdoc, and of course, the core facility at Xavier, who have done many of the bioassay studies for me. Thank you.